Good evening friends, you are most welcome to Think Eyes channel. Today we are going to discuss Practicing Anthropology by MAAN IGNOU, and our topic is Advanced Ideas in Practicing Anthropology, in which we are going to discuss application of genetic polymorphism. Let us begin the topic. DNA profiling. DNA profiling is a way of identifying a specific individual rather than simply identifying a species or some particular trait. It is also known as genetic fingerprinting profiling. As a technology, it has been around since 1985 when it was announced by its inventor, Sir Alex Jeffrey. DNA profiling is currently used both for identifying paternity or maternity and for identifying criminals or victims. DNA uses a specific type of DNA sequence known as microsatellite to make identification much easier. Microsatellites are short pieces of DNA which repeat many times in a given person's DNA. In a given area, microsatellites tend to be highly variable, making them ideal for DNA profiling. By comparing a number of microsatellites in a given area, one can identify a person relatively easily. DNA profiling has a high success rate and a very low false positive rate, making it an extremely popular form of paternity and maternity verification. In forensics, DNA profiling is very attractive because it does not require actual fingerprints, which may or may not be left behind and may or may not be obscured. Because all of the DNA sections are contained in every cell, any piece of a person's body, from a strand of hair to a skin follicle to a drop of blood, may be used to identify them using DNA profiling. This is useful in the case of identifying a criminal because even a drop of blood or skin left at the crime scene may be enough to establish innocence or guilt, and it is virtually impossible to remove all physical trace of one's presence. DNA profiling is useful in the case of identifying victims because even in case where the body may be disfigured, the disfigured past identification and teeth or other identifying features may be destroyed. All it takes is a single cell for positive identification. Summary: the polymorphism occurring while gene replacement is in process of transient, since as soon as the favored allele is fixed, the population becomes monomorphic. However, many characters in human populations are more or less permanently polymorphic. One of the most obvious examples of such balanced polymorphism is sex. Others that occur in many human populations affect the blood group systems, secretor status, hemoglobins, red cell enzymes, and serum proteins. Non-coding regions of DNA from homologous chromosomes pairs also demonstrate an average of one nucleotide difference for every 250 nucleotide sequence. The nucleotide differences are called polymorphisms. Humans have such extensive polymorphism that each person's DNA is an individual as his or her fingerprints. These polymorphisms then become genetic markers that can be traced within members of the family just as genes are traced. The role of genetics is emerging as an increasingly important aspect of health care. A number of RFLPs associated with human disease are now being used in prenatal diagnosis and to determine delayed onset disease and dominant and recessive carriers. A number of RFLPs have now been cloned. Those for cystic fibrosis, Duchenne's muscular dystrophy, sickle cell anemia, thalassemia, hemophilia A, and Huntington disease, etc. Information from a genetic test can be used to diagnose disease, identify risk as for future disease, and predict treatment response. Genetic polymorphism provides us with the ability to predict inter-individual differences in susceptibility to clinical disease. Biomarkers of susceptibility include polymorphism in drug or carcinogen metabolism, in DNA repair capacity, and in genes that control cell growth. Wide variation in drug or carcinogen metabolism have been widely investigated and clearly shown to be an important determinant of individual cancer susceptibility and adverse drug reaction. Such polymorphisms in drug or carcinogen metabolizing enzyme may be due to heritable and or to environmental factors, and the modern application of metabolic phenotyping and genotyping methods to epidemiological studies has provided new insights into such gene environmental interactions. Polymorphism in DNA repair or processing of DNA damage have long been evident from rare hereditary disorders involving defective DNA repair or chromosomal stability. Today, about 130 different genes have been shown to be involved in DNA excision or base repair, and polymorphisms in gene-specific DNA adduct repair have been correlated with biological outcomes, mutations, drug sensitivity. Moreover, lower DNA repair proficiency has recently been associated with increased susceptibility to cancer of the skin, brain, lung, stomach, breast, bladder, head or neck, and colon. While over 100 genes have been identified that serve as positive proto-oncogenes or negative tumor suppressor genes, regulators of cell growth, as well as the cell cycle and apoptosis. Examples: cyclins, cytokines, chemokines, caspases, etc. These have been largely associated with rare hereditary disorders involving greatly increased human cancer susceptibility. Besides, the advent of DNA technology has gained wide acceptance in criminal offenses involving biological evidences such as murder, sexual assault, in case of mass disaster, paternity disputes, in identification of mutilated bodies and exhumed skeletons. Anthropology is on the verge of becoming a purely applied science. Advanced scientific and technical know-how on the Human Genome Project are enabling us to understand the rich genetic resources India possesses. Advanced scientific and technical know-how on the Human Genome Project are enabling us to understand the rich genetic resources India possesses. India, with diverse population, has incredible scope to move forward in the field of genetic epidemiology and to develop effective strategies to prevent both infectious and complex lifestyle diseases like type 2 diabetes, obesity, hypertension, cardiac disease, cancer, and others. Thank you very much.